All right, welcome to the first lecture of the first unit of the year. This is going to be on the scientific method and how we can think like a scientist. Um, with this first lecture, as you listen, you're going to be answering questions that go along with the lecture. I'm going to be providing an example to you as well. When you come into class on Thursday, we'll read about this topic, so you'll have to bring your textbooks to class on Thursday. All right, so to get started here with the scientific method and thinking like a scientist, we got to first think about what the scientific method starts with. So let's look at an overview of that right now. The scientific method is really just an instrument that scientists use to question things around them in the world. Okay, scientists observe different things happening, and then through that observation, ask questions about how different things are connected in the natural world and in the universe too. So as we ask questions, then we have to design experiments to figure out or try and figure out the answers to those questions, and that is what the scientific method really is. So in general, we're going to ask a question first, then develop a hypothesis, which is an educated guess. Then we're going to test our hypothesis through experimentation. And then we're going to draw some conclusions. And most often, scientists will draw a conclusion that is, we have to design another experiment so we can answer another question that our experiment gave us. Okay? So that's a quick overview. Here is the overview kind of in a picture or diagram format. I really like diagrams, so we'll use these a lot during classes here. This is not going to be an outline that every single scientist uses. Not everybody needs to start at asking a question, but for the most part, scientists will do that. So you'll see and identify a question that you want to answer. You'll see what have other scientists done. You'll then construct a hypothesis, which is, again, an educated guess based on the research. You'll test that hypothesis with an experiment, and then you'll analyze your results. Once you analyze your results, you'll see if your hypothesis was supported or not supported. And if it's supported, then you can report your results in a journal. A very famous journal is called Nature, okay? And they'll publish your results and tell other scientists what you have done. Even if your hypothesis, though, turns out to not be supported, you can still report those results in a journal. Okay, every result can be a significant result. But if your result turns out to not be supported, you might want to think about trying again, making a new hypothesis, and then testing your experiment again. So that's a little overview of the scientific method. So first thing we're going to start with is asking a question. So what does asking a question entail? Well, they're going to start with looking at something that they do not understand and asking a question on how we can get at that. So let's see. For you, we may look at some kind of plant. Here's our soil. I have our little plant here. All right, so here's our flower. And what I or you may want to test is what's going to make this flower grow the best or grow the tallest, okay? So you may see, well, oh, here's one flower that is tall, okay? And maybe we have another flower over here that is not quite as big as the other flower. You may ask yourself, why did that occur? I don't understand. What's the difference? Why did this flower get or become taller than this one? Okay, or that plant become taller than this plant? So that would be a question or an observation you could make, and then asking that question could be the start of your experiment. The next thing we're going to be doing is developing a hypothesis. Okay, now again, a hypothesis is an educated guess or a possible answer, not just a guess. So when I ask you to create a hypothesis, I'm not saying, hey, just think about it for a second and make a guess. You really got to think about it hard, you know. Analyze what you already know about the topic, and then put forth that educated guess based on what you know already. Okay? So again, this relates to the observation and the scientific question. So if we were looking at our plant experiment, we would say, maybe the amount of nutrients in the soil in this area is at a higher amount than the amount of nutrients in the soil in this area. And that may be what has led to the difference in height. So if we were writing that hypothesis, it would be something like, if we give more nutrients to a plant, then it will grow taller. Okay, so that would be our hypothesis. Educated guess based on what we know about plants. All right, moving on. We then design an experiment. The controlled experiment is really important for testing a hypothesis because you got to really know what is affecting the plants. All right? So in our control group, well, in a controlled experiment, you divide up your experiment into two groups. One flower that's going to receive no treatment, and then another flower that will receive treatment. Okay? And that's basically it. Okay? For every controlled experiment, you've got to have two groups, the control group and the experimental group. And that's how we're going to keep our experiment from becoming too large, because the more variables you have, the more groups you have, the less likely it is you're going to figure out what is actually happening. Okay? So if we go back to our example here, we'll have our control group. Let's go to another slide here. All right, so in our control group, so let me just let me erase that. All right, so there's pot 1. And here will be pot 2. Sorry, I'm not the best drawer. Get rid of this. Okay, perfect. So here are my two groups. And in one group with the soil, maybe I'm going to put a higher mixture of nitrogen, phosphorus, and potassium. Okay, so we'll do nitrogen, phosphorus, and potassium. I'm going to put a really high amount of that fertilizer in the soil. Okay, this will all be fertilizer. And in the other one, let's say I'm going to go out to the environment I'm in, and I'm just going to put a regular amount of soil in there. I'm not going to adjust the amount of fertilizer. So in this one, I have regular soil with extra fertilizer. And in this one, I just have regular, in this one over here, I just have regular soil. Now we're going to plant some seeds. And we're going to see how each one does. All right, so that'll be our experiment. If we were to look at this, this would be our experimental group because we have changed something. We've altered the experiment. And here, this will be our control group. Our control group is going to have nothing altered, so I didn't add anything extra. We're just keeping it the same as it is in its normal environment, and we're going to see what happens. All right, back to this. With our design and creating our groups, we're going to have variables, okay? And ideally, there's going to be one variable that you change and one variable that you measure. The independent variable is the variable that you change. Okay, so you, as the scientist, are going to go in and change this about your experiment. With the dependent variable, it is always the item or thing that you are measuring in your experiment or counting. Okay? All right, let's go back to our example then. If we were to think about this then, get this out of the way. I'll rewrite this over here somewhere. Be our control. If we were to analyze this then, our independent variable would be something that we changed. So that would be the amount of fertilizer. Okay, that's what we have added into the pot. We changed that. What are we going to measure? Well, we're going to measure plant growth. We're going to see if that has an effect on the plants. Okay, the fertilizer, we're going to see if the fertilizer has an effect on plant growth. All right? Fantastic. After we do our experiment, we're going to collect our results. And when we collect our results, it's going to be in the form of data. We're going to have to make observations and measure our plants to collect our data. We're then going to see if our data is supported by our hypothesis. So let's pretend that 3 weeks go by. Whoa, look at that. Sorry about that, guys. All right, let's pretend that after 3 weeks, this flower has grown. Let's say this right here is 5 inches. Okay. Actually, you know what? In science we use centimeters, but I'm just going to use inches to simplify the measurements right now. Okay. So we'll do 5 inches there. Let's say in our experimental pot, after 3 weeks, our flower only grew 3 inches. Well, you might think, wow, I gave this plant more fertilizer, and it didn't grow as tall as the control sample or the control flower. So you might ask yourself, wow, why did that happen? And that would be part of your next experiment, okay? The results are not always going to be what you expect them to be. And if this is the way it turned out, then this is the way it turned out. All right. So our observations were that the control plant grew 5 inches and that the experimental plant grew 3 inches. And now we're going to see if our hypothesis has been supported. Go back to our previous hypothesis. The hypothesis is if we give more nutrients to a plant, then it will grow taller. Was that supported? No, it was not. Too bad. Wonder what happened. All right, to finish off here, now we draw our conclusions. Okay, once we have gathered our data and we've reviewed it, so we've looked at the measurements again, we have to draw our conclusions. Something, either whether it's the fertilizer inhibiting or stopping the plant from growing as tall, or maybe it's some other factor that when we set up the lab or set up the experiment, we didn't think of. Maybe we put them in a weird spot where they got alternate or different amounts of light, and we didn't even realize it, right? Or maybe it's the way we packed the soil together. Maybe the packing of the soil didn't allow the plants to germinate as fast enough. So there's a lot of different things that could have gone into our setup. Maybe it's that the fertilizer, if you increase the amount of fertilizer, the plant actually is harmed. That could also have been an effect, all right? What we're going to have to do now is, after collecting our data and determining that we are rejecting our hypothesis, we're going to have to form a new question based off what we learned. Well, fertilizer didn't happen to help the plant grow, okay? So maybe it's that the differing amounts of light affect whether plants grow taller or shorter. Or maybe it's the packing soil you want to test. Maybe it's the size of the pot. All of those things can be tested in your new question. And then when you go back here, you design a new experiment and test it again to see if the results you get match what you thought you would have gotten. Okay? All right, so that concludes our first lecture on the scientific method. On Thursday, we're going to be reading about this and using some examples to help illustrate kind of what we're talking about with the scientific method, but I really want you to remember, look back to this picture here. Scientific method does not always start at the top and go to the bottom. This is a fluid model, so things can go back and forth. Maybe you test something and you realize the results aren't what you expected, so you go back and you construct a new hypothesis and test, and then maybe as you're testing, you realize you need to do some more background research, so you go back here and do more research. All of this can happen in a scientific experiment or a research study, okay? And that's the beauty of science. You ask a question, and that question will lead you to an answer that could then generate more questions. So we constantly are learning about the world around us and how it's connected to us. All right, nice job. I will see you in class tomorrow, and thank you for listening.